Growth and multiplication of bacteria. Bacteria divide by binary fission. When a bacterial cell reaches a certain size, it divides to form two daughter cells. Nuclear division is followed by cell division. Generation time. The interval of time between the two cell divisions, or the time required for a bacterium to give rise to two daughter cells under optimum conditions, is called the generation time or the population doubling time. In Escherichia coli and many other medically important bacteria, the generation time is about 20 minutes. Some bacteria are slow-growing. The generation time in tubercle bacilli is about 20 hours. In lepra bacilli, it is as long as about 20 days. Batch culture and continuous culture. When bacteria are grown in a vessel of liquid medium, multiplication is arrested after a few cell divisions due to the depletion of nutrients or accumulation of toxic products. This is a batch culture. By the use of special devices for replenishing nutrients and removing bacterial cells, that is chemostat or turbidostat, it is possible to maintain a continuous culture of bacteria for industrial or research purposes. When bacteria multiply in host tissues, the situation may be intermediate between a batch culture and a continuous culture. The source of nutrients may be inexhaustible, but the bacteria have to fight the defense mechanisms of the host. Bacteria growing on solid media, for example, blood agar and McConkey agar, form colonies. Bacterial growth may be considered at two levels: increase in the size of bacterial cell and increase in the number of cells. Growth in numbers can be studied by bacterial counts. Total count and viable count. Two types of bacterial counts can be made: total count and viable count. It can be done by various methods. For example, direct counting under the microscope using counting chambers. The total count gives the total number of cells in the sample, irrespective of whether they are living or not. The viable count measures the number of living cells, that is, cells capable of multiplication. Viable counts are obtained by dilution or plating methods. In the dilution method, the suspension, whose cell count is to be determined, is serially diluted. The dilutions are made to the point beyond which unit quantities do not yield growth when inoculated into suitable liquid media. Each dilution is inoculated into the respective tubes containing liquid media. The viable count is statistically evaluated from the number of tubes showing growth. This method is not accurate, but is used for the estimation of presumptive coliform count in drinking water. The presumptive coliform count is a method of estimating the level of pollution of drinking water. In the plating method, cereal dilutions are dropped on the surface of dried plates and colony counts are obtained. The method commonly employed is that described by Miles and Misra, 1938. The number of colonies that develop after incubation gives an estimate of the viable count. Bacterial growth curve. When a bacterium is seeded into a suitable liquid medium and incubated, its growth follows a definite course. If bacterial counts are made at intervals after inoculation and plotted in relation to time, a growth curve is obtained. The curve shows the following four phases: lag phase, log or exponential phase, stationary phase, decline phase. Lag phase. Immediately following the seeding of a culture medium, there are no appreciable increase in number, though there may be an increase in the size of cells. This initial period is the time required for adaptation to the new environment. The necessary enzymes and metabolic intermediates are built up in adequate quantities for multiplication to proceed. The maximum cell size is obtained towards the end of lag phase. The duration of the lag phase varies with the species, size of the inoculum, nature of the culture medium, and environmental factors such as temperature. Log or exponential phase. Following the lag phase, the cells start dividing, and their numbers increase exponentially or by geometric progression with time. If the logarithm of the viable count is plotted against time, a straight line will be obtained. In this phase, cells are smaller and stain uniformly. Stationary phase. After a varying period of exponential growth due to depletion of nutrients and accumulation of toxic products, the number of new cells formed is just enough to replace the number of cells that die. Equilibrium exists between the dying cells and the newly formed cells. So, the viable count remains stationary. In this phase, cells are frequently gram variable and show irregular staining. Sporulation occurs at this stage. Decline phase. This is the phase when the population decreases due to cell death. Besides nutritional exhaustion and toxic accumulation, cell death may also be caused by autolytic enzymes.